Hello, and thank you for joining us for today's webinar hosted by the ISEH. My name is Novella Guidi, and I'm a postdoc in the School of Gerontology of the University of Southern California in Walter Longo's lab. Previously, I earned my PhD in molecular medicine and stem cell aging at the University of Lund under the mentorship of Professor Hartmut Geiger, one of the speakers of today's webinar. If you have any question at any time during the presentation, please type your question in the question box or chat box. We will have time for questions at the end of the webinar, but you are welcome to enter your question into the chat box at any time during the presentation. So now it's my privilege to introduce our first presenter for today's session, which is Hartmut Geiger. Hartmut Geiger obtained his PhD at the Max Planck Institute for Immunobiology at Freiburg, Germany. His postdoctoral work then took him to the University of Kentucky in Lexington. And in 2002, he was appointed as assistant professor in the Division of Experimental Hematology of the Department of Pediatrics at the Cincinnati Children's Hospital Medical Center. Hartmut was in 2008 appointed as a research professor and ultimately in 2013 as a director of the Institute of Molecular Medicine at the University of Ulm, while still holding a dual appointment with Cincinnati Children's, now at the level of full professor. So please, Hartmut, we are looking forward to your presentation.
Good morning, everyone, or good afternoon, no matter where you are, and it's a real honor to be part of the webinar today. And the title of my presentation is Rejuvenation of Stem Cells, a Road to Healthy Aging. Hope everybody can see that. So, usually I do some introductory slide. Why should we be interested in aging? Because most of you on the other side might be 30 years or less or 50 years or less, but definitely not aged yet. And this is always a graph I like to show. This from the World Health Organization. It's the number of people shown here and the age here in years here. And we're currently here at 2018 roughly. And this is divided in less developed countries and more developed countries, as you see, and in the people 60 plus, people 12 to 24. So in developed countries, Europe and the US and Canada, all of that, you have a linear increase of people 6 plus. And we have a decline in people 12 to 24, so if we don't make enough babies, that's a different story. The more interesting part comes also from what at that time they called less developed countries. I guess they mean primarily India or China, with some I don't share the less developed. But the interesting part here is that you see that an exponential increase in the people 60 plus, and you're in the start getting exponential right now. So the relative change over time in people getting older is much more dramatic in these countries, and it usually goes along with that aging-associated diseases that might have to do with aging of blood stem cells also increase exponentially, which means at the end we might end up with an exponential times exponential combination, which is not really a good thing. So in science and research, what we should do is then identify molecular and cellular mechanisms of aging that cause age-associated tissue dysfunction diseases, and hopefully we can develop innovative therapies based on that that might lead to healthy aging. Now, as you know, we are working with hematopoietic stem cells, and that's the topic of our talk today. And this is a very simple slide summarizing research from many, many laboratories all over the planet that worked on aging of hematopoietic stem cells. And I think the bottom line everybody agrees to is that you have young stem cells in a certain number in the bone marrow, and that gives what we call a balanced output towards myeloid, lymphoid, and also erythroid differentiation. If you look now at aged stem cells in aged animals or aged human beings, the first thing we see actually is that the number of these stem cells is not decreasing, but it's increasing, at least the phenotypic ones. And they tend to do better myeloid differentiation output and reduce the lymphoid output and also the erythroid output. That can be driven by intrinsic and extrinsic signals. That's also clear. So that might be signals coming from stem cells themselves and from the niche. And they might be linked to aging-associated diseases. And I have two examples today for intrinsic or extrinsic parts of that. Now, the title of our talk today is about rejuvenation. That implies that something can be reverted, or at least attenuated. If you think about it, it's only possible if the mechanism that leads to aging is also reversible. You've probably heard a lot of theories on what causes aging. So there's sunlight and radiation, which means it's called DNA damage directly. Or you have telomere attrition. You've probably heard about that too. That means telomeres get shorter with aging. And I would call them irreversible factors of aging, because there's almost no way that we can get this back no matter how hard we try. You probably also heard about it. There is epigenetic changes as shown here, methylation patterns of DNA, acetylation of histones and so forth. Then there are changes probably in the ribosomal translational machinery, or there's changes in mitochondria. All of them are in part reversible because they're all protein-based. Usually, with the exception of mitochondrial DNA. But even there, you might find ways to replace damaged ones with not damaged ones. So a lot of them might be more reversible. So that's why we a long time ago started to initiate to check how much is being driven by reversible or irreversible mechanisms. And what we initially did in an experiment was very smart. So we took stem cells from a young mouse, bone marrow, and from an old mouse. We did what we call competitive transplants, equal numbers of these cells into a radiated lethally mouse. Then we waited after the hematopoietic system were constituted, and then we just let the animals go or irradiate it again, so put more damage into the system, and then measure the chimerism in the hematopoietic system 3 months later again. So the assumption would be if one over the other population, young or the aged ones, would be more susceptible to DNA damage and irradiation, you would see a change in chimerism over time before and after irradiation of these groups. And here we only show the chimerism driven by the old bone marrow, at the end and old stem cells. And what you can see in all hematopoietic tissues, there's different level of chimerism, but the chimerism does not change after radiation. It says that it stays exactly the same. That would mean the young and the old system suffer identically upon another level of DNA damage, which is highly interesting. So reports would have said the aged ones are more susceptible to irradiation or DNA damage in vivo. And then we checked mutation frequencies, which is a small blue indicator mouse, and it would take too much time to explain that in detail, but it will ensure you that it can detect point mutations, deletions, and translocations in hematopoietic cells. And this is a richer mutation frequency. What you can see here is that young animals and old show a difference, but it's not dramatic. It's probably twofold at steady state. Now, if you irradiate the animals and then measure the recovery in young animals, you actually see that the mutation frequency that we finally find in bone marrow is actually not increased. It's actually going down compared to the young initial level. And even in the aged ones, that's even more important, this also goes down dramatically. So upon another level of DNA damage, what you see is not an increase in mutations in the system, but a decrease. So that strongly suggests that DNA damage by itself is probably not, or mutations upon DNA damage is probably not the major driving force that accumulates upon aging or will contribute to these aging phenotypes of stem cells. So then we focused a little bit more on protein biology and looked at something that I would refer to as order and disorder of stem cells. So a lot of phenotypes that you see in aged stem cells could be associated with changes in distribution of proteins in stem cells, but nobody has measured that so far before. And what we have is described as a polar distribution of protein in stem cells or apolar distribution of protein in stem cells. Polar means there's a certain order, either in the cytoplasm or the nucleus. Or apolar means there's no order. It's a little bit more random, the distribution. So then we sorted out hematopoietic stem cells from a mouse. And in our case, they're lineage negatives, CAR positive, KIT positive, FLIC2 negative, and CD34 negative. And we did that for young stem cells and aged stem cells. And we did immunofluorescence on them and checked for markers that are associated with polarity. CDC42 is such a marker, tubulin can be a marker, and pericentrin is such a marker. You can easily see that young stem cells are very polar for these markers. For example, for the cytoplasmic marker CDC42, and aged stem cells are apolar for this marker. And you can further quantify that. So you have the percentage of polarized cells here. If you're young, white bars, you see that 60 to 70% show polarity. In the aged ones, only 20%, so means 80% are apolar. And the same is true for tubulin and all other markers that we tested. And that just shows you here, even if you're in young polarized cells, CD42 usually goes together with pericentrin. And even in the aged ones, if you have polarity, small level like here, for CDC42, pericentrin is not polarized either. So they're pretty disorganized. So if we then ask next, what is the mechanism for this polarity-apolarity? And it turns out that polarity has been shown in other systems to be regulated by a small road GTPase, CDC42, which in itself is a polarity marker. And as all road GTPases, its activity is not determined by the expression level, but by the loading with GTP. So when CDC42 is associated with GTP, then we can interact with effectors and then regulate DNA synthesis, apoptosis, adhesion, cytoskeletal reorganization, aka polarity and migration. And you have exchange factors like GAP and GAFs that are responsible for the amount that's unloaded or loaded with GTP, and all of these factors are regulated by ligand receptor interactions or intercellular interactions. So the first thing we tested then is what is the activity of CDC42 upon aging in various tissues, and because you need to measure activity and not expression, we need to have a PAC effector pull-down assay in which we pull out CDC42 that's GTP bound by an effector protein called PAC that is bound to this road GTPase. And in that case, it can interact with the GTP bound protein. Oh, there's a rescue helicopter here, I apologize. And then you can compare the total amount of the GTPase put on a Western. And then you have the C42 GTP bound form listed here, and the total amount listed here, and this is normalized to beta-actin. And what you see right away is that upon it aging, the activity actually goes up. It's not going down, it's going up. And you can see that in various levels between 2 and 3-fold higher upon aging. And this is not restricted to the hematopoietic system only. You can see multiple tissue of aged mice. And we have shown that the elevated activity, if you do artificially get CDC42 activity elevated in young mice, this results in premature aging of stem cells. So this implies that the elevated activity is causative. So this then lets us to think about a pharmacological intervention. If this is causative, then we might find an inhibitor of CDC42 activity, which we actually get via collaborators in Cincinnati, and that is called casein. What casein is doing, it inhibits the dissociation of GDP from CDC42, so it keeps it longer in a GDP form and therefore reducing the activity because GDP cannot be loaded. And that's easily shown here. So if you look at the activity again of CDC42, it's set to 1 in young animals, and then it's going up to 2 to 3-fold higher in the aged animals. If you now look at stem or progenitor cells that have been incubated with casein at 5 micromolar, you see a drop to the level seen in young animals if you incubate aged stem and progenitor cells with casein. And this is just a verification with Western blot. You can easily see that here, young certain level aged up, and casein reduces this to a normal level. So then we ask also if it reverses this aging-associated apolarity in stem cells, because that's the first phenotype we had for aging. And you can see if you change the activity of CDC42 in aged stem cells, you get a repolarization of the stem cells for the markers CDC42, for the markers tubulin, for the markers perissensin, and so forth. And you can actually quantify that, and you have a dose-response curve. So you can see that in the young ones, again, high level of polarity. In the aged ones, you lose this polarity. And if you titrate in casein, you gain this activity, polarity back. And at 5 micromolar, you have it back to the young level. Good. And now I'll cut this very short. We did a lot of functional assays, transplantation assays, homing assays, adhesion assays, and so forth. And whenever you look at the function of the stem cells that got treated with casein, so that reduced the CDC42 activity to levels seen in young animals, they looked very similar to young stem cells, but very distinct from old stem cells. So it's fair to say that reducing CDC42 activity to the level seen in young stem cells rejuvenates aged stem cells. And it's been published. Now, I would like to talk a little about, as I said, there's intrinsic factors and stem cell extrinsic factors, because we all know that stem cells are not living alone. They're like their niche. They might even have other stem cells or hematopoietic cells that can influence them. So they're living in a very complex social structure. So we were asking initially, so are there any factors in the niche that might change over time in aged animals? And first work has been done a long time ago, actually, by Peggy Godell on this work. And part of osteopontin has also been published, looked by in stem cells by Susan Nielsen in Australia. So what we did is looked at what kind of cytokines are differentially found in young versus aged stroma, initially in vivo, and one thing popped up right away, osteopontin, because it was the only one that's actually, from the ones we tested, it's actually been reduced in aging stroma. So you can see this is the secretion in vivo, it's down. Then we looked at RNA expression in young versus aged stroma cells, that the expression is down, and this is not seen, for example, in differentiated bone marrow hematopoietic cells or even in stem cells. So the huge difference is coming from the stroma. And it turned out that the major part is actually coming from osteoblasts because they are OPN. So first part is that the number of OPN positive cells also reduced dramatically upon aging, and it's primarily driven by osteoblasts. They show the clear reduced mean fluorescence intensity for osteoblasts, for OPN upon aging, but it's not mesenchymal cells, CAR cells, or CD31 endothelial cells. So that would mean there are osteoblasts that secrete less osteopontin that confers this lower level of osteopontin in the stroma. Good. And then I forgot to mention that we could clearly show also in transplantation experiments that this lack of osteopontin leads to premature aging of hematopoietic stem cells, also by most likely modifying CDC42 activity. So it might be one connector where you have the extrinsic signal going to the intrinsic signal. Now, then we tried very hard to reproduce the data initially in vitro in cell culture experiments. So we gave osteopontin back to H stem cells and said, yes, maybe that's working. And it turned out it does not work because we forgot some small point, which is that osteopontin only works in vivo after it has been cut by thrombin. You can see here, this is the endosteum here, and you can see osteopontin in the green part. It's close to the endosteum with a lot of osteoblasts, and thrombin is really close by, and it means in vivo you have osteopontin always close with thrombin. I mean, if you look at thrombin, osteopontin that you buy from the store has a mass of 75. If you put it together with thrombin, then you have a lot of truncated versions of osteopontin. It's cut down by thrombin. And it turns out that only the cut form down here, can you see, is able to make these H stem cells polarized again. Again, most likely via changing CDC42 activity, but this full-length thrombin is not acting on H stem cells. So it's highly interesting that only a cut form, cleaved form, is able to have a biological activity, a very prominent one, repolarizing H stem cells and maybe making them functionally younger. So what we did then is we further purified these osteopontin fragments that you see here and get certain via HPLC and had fractions A, B, C, D, and E, and looked at their active in vitro assays and test the function of stem cells. So then we took osteopontin fractions, C and D, incubated then H stem cells with these fractions, and tested in transplantation assays whether these stem cells also function younger compared to their old counterparts. And it turned out that only this very fragmented D, so special cut thrombin fragment, was able to reduce the number of stem cells in transplants and also repolarize the stem cells for additional set of markers. These are epigenetic markers in the nucleus, which are similar to the CDC42 marker I just talked about for the other hereditary phenotypes. So it means that there are multiple ways to make stem cells younger. For example, CDC42 activity is also driven by elevated wind intrinsically to stem cells, but it's also changed about lack of osteopontin, stroma osteoblast. I showed you two examples where we can intervene, kind of pharmacological way we can add osteopontin, and we can inhibit by casein and make the stem cells younger. So, but what is rejuvenation of stem cells good for? I have 2 more minutes to talk about that. So far that's a good academic exercise, but there's a real-life problem called aging-associated immune remodeling that results in increased susceptibility to infections, increased onset of cancer, decreased responsiveness to vaccines in the elderly, and increased onset of autoimmune diseases. And so far people have associated with that thymic involution, so the thymus gets smaller when we age, become older, or chronic viral infections like CMV. And we thought maybe all of that has also to do with aging of stem cells that have changed their function upon aging. So what we did then is a very simple assay in which we transplanted stem cells, purified stem cells, from young and aged animals into donors, and we're asking as a recipient, we took rat mice, and we're just simply asking if you have these stem cells transplanted, do they form a young or aged immune system? So what you now have is the donor is young or the donor is aged, and you transplant them into these RAG1 animals. What you can see is that if you transplant again stem cells from a young donor, they can restart the thymus in RAG1 animals, which have a very small thymus due to the lack of T and B cell development that they initially have. But the H stem cells, although they're exposed to the same environment, same thymus, cannot restart the thymus. And this is also reflected when you just check young and aged animals. So that means you have information on reconstitution ability already stored in the stem cells. Now we've asked whether the vaccination response might be restored. So we use the DNA vaccination with the hepatitis antigen, which is a very established method to measure immune response in mice by the generation of a special antigen that's stored on CD8 T cells that then could be activated against the antigen. So what you need to focus on is this bar here, this area, which tells you whether the mouse is immune competent after the vaccination. Now, if you look at young mice, if you do that, they have a certain level of immune competence. The aged one, it's really gone. Now, if you do that if you just transplant young stem cells, you get also the same immune competence. And if you transplant just aged stem cells, you also lose this immune competence, which means, again, the immune competence to respond to this antigen is already somehow stored in the stem cells because that's the only thing we transplanted into just recipients. And then you can ask the same question if you make these stem cells younger, rejuvenate, again with casein, as I've shown before. So they performed much younger in transplantation assays. You can ask, if you do this in the immunization again, do these aged stem cells, when they're treated with casein in the transplantation assay, and then later on in the immunization assay, do they respond now to the vaccination? And they do, and you get a very nice vaccination response back, although you transplanted with aged stem cells, but again rejuvenated aged stem cells. And you can see that in the spleen and also in the liver. And if you remember, this number was 6, it's just the young ones, so you get almost a perfect immune response back. That means rejuvenation of stem cells can restore, under these conditions in a rat mouse, a perfectly young immune system with a strong response to immunization, which is, I think, a very interesting achievement that would bring me to the end that we think that aging leads to disorganization in the stem cells with a lot of components within the cell that is usually protein components, which is reversible because you can restore this order by different various pharmacological approaches. And at the end, I would thank, first of all, the funding institutions that allow us to do that, and especially Carolina, our junior group leader in the group, that's done a lot of work on the polarity study. Then Novella, as you heard about, a lot has done on the OPN story, and Hannah, which has done a lot of work done on this immunization study. Hannah is here, and all the other lab members that contributed to that, and Bettina, which has mostly done the studies on the DNA damage. And this is only possible with very strong collaborators all over the world. And with that, I would like to stop here and head over to Novella.
Thank you so much, Hartmut, for a great talk. And I want to remind everybody, all the attendees, that if you have any question at any time during the presentation, please type your question in the question bar or chat box you see on your screen, and we will have time for questions at the end of the webinar. So let's go ahead and with our second speaker of today, which is, I'm pleased to introduce, is David Bryder. David Bryder obtained his PhD in 2003 at the Stem Cell Center at Lund University in Sweden. After this, he did his postdoctoral work at Stanford University, and in 2006, he returned to Lund University to establish his own research group, first at the Department of Immunology, and from 2014 as part of the newly formed Division of Molecular Hematology. David was recruited to Gothenburg University at the Sahlgrenska Cancer Center in 2017, but currently also maintains an affiliation at a laboratory at Lund. And he is a full professor at both his current affiliations. So thank you, David, for being our speakers of today, and please go ahead with your presentation.
So thank you for that. and I would like especially to start by thanking ISEH, and particularly then Novella and Tamara, for the opportunity to share some of my personal views on the study of aging hematopoiesis, with a particular note and on the prospects for restoration of stem cell function. Now, in contrast to Hartmut, I will try to maybe not be so mechanistically oriented in my talk, but rather outline, the foundations that, this concept of hematopoietic stem cell aging or stem cell aging for the organism. So what are they actually based on, and are we always correct when we are discussing sort of an organ system aging from the perspective of stem cells? So coming from Sweden, so this is a Swedish car, and the thing with these two cars is that actually they are photographed at the sort of same age, but they can manifest, of course, quite different. So you could sort of appreciate perhaps that the car to the left there would not be a functioning car. The car to the right would be, at least by appearance, it would look, well in order. So I think that, so here is the first key definition I'd like to introduce, and that is that between chronological aging and functional aging. And so I think that looking at a lot of research that's going on in this field, so sometimes I think there is a confusion as to whether chronological aging actually translates into functional aging. And I will see if I can present and convince you of some experimental approaches where one can approach this. The second definition or concept I'd like to introduce is that of segmental aging, which I think is more established in the aging field. So what we mean with the concept of or the term segmental aging is really, that in an organism, so not all our organs are aging at the same pace or same speed. So you can, for instance, imagine that you can have aging of the brain, for instance. It could be a very late event, aging of some type of organ or tissue that arose maybe earlier in fetal, more with a fetal association, would manifest earlier on. Now, this concept, of course, you can imagine would also be one that you could apply to within an organ system. And so I think that Hartmut was touching on it a bit in the end of his presentation with the immune system. So we can, for instance, imagine that our blood systems would, we would have manifestations, for instance, with immunity, that at the sort of end stages of life, that let's say the immunity, the arising in response to an antigen, that we would acquire that early on. We could lose it with age. We would acquire sort of some type of immunity later on, or perhaps we would not acquire it. So the concept of segmental aging is not only between organs, but it actually can apply to different types of cells within an organ also. So now I will not go over in great detail all the sort of mechanisms or broader mechanisms. I think there are many excellent reviews on this topic. This one in particular, published a few years ago now, on the hallmarks of aging, so in which the authors listed 9 fundamental sort of aging mechanisms. And looking at these mechanisms, so we can have certain mechanisms that we can consider reversible, sort of from the mechanistic point of view. Hartmut was touching on this also, that for instance changes in DNA or in telomere shortening, for instance, so those would probably be more difficult, perhaps not impossible, but at least more difficult to correct. Whereas, of course, other changes that are more gradual, not based on more on sort of stable changes, perhaps are mechanisms that will be easier to counteract. Now, the concept that DNA mutations would be associated with age, we would have to say is not a new idea, and for sure it holds true also for many organ systems. And I would say for the blood systems, it has become quite apparent during the latest years that we have the phenomenon of clonal hematopoiesis, which probably has a root cause, at least to some extent, at the stem cell level. Now, our interest in this was really not to sort of refute that, but rather to see whether changes like that were the only or dominant drivers of sort of hematopoietic or stem cell aging. Now, of course, talking about these sort of aging effects in hematopoiesis, so I think that it's important to sort of try to define what is the starting point of this, and I've tried to here to just outline. So hematopoiesis, where hematopoiesis occurs actually from coitus and until advanced adulthood, and so prior to birth, we can see that of course hematopoiesis shifts quite dramatically in terms of sites. And then following birth, of course, we have most of the hematopoiesis going on in our bone marrows. Now, the shifting production sites, of course, is likely of importance if you consider like this interaction points of the cells, and to some extent, of course, that can guide the potentials of the cells, but also there are other adaptive changes. So in up to the right here is just a sort of depiction of the different types of globins and when they are produced in relationship to age. And so this is probably then a reflection of more adaptations that actually, during development, we have a need to also sort of sort of correct or adjust the types of cells and type of effector functions to the phase in life. Now, if we look on this figure down to the right here, so this is really a depiction then of what we talk about with much of what Hartmut and me have been talking about in terms of adult hematopoietic stem cell aging. And so, of course, we have the different waves of hematopoiesis, so not all our blood cells are actually derived from what we sort of define as definitive hematopoietic stem cells. And you have different phases also as to definitive hematopoiesis in terms of changes in function. So, at some point in time we have these type of embryonic definitive hematopoietic cells that produces some quite unique developmentally restricted immune cells, for instance. So really what we've been, a lot of the focus in the field I think has been on is on actually the changes that occur from birth and onwards in the stem cell compartment. Now I deliberately here chose to display some graphs of human, timelines from a human perspective versus that of a mouse, because a lot of these studies are actually made in mice, but of course, as you are aware, there's quite a lot of differences in lifespan here. So actually, what we would say a newborn human would actually, in terms of chronological age, be something even of an intermediately aged mouse. Now, that difference aside, though, there are conserved changes in between these two systems, and so here is just some work that Alexandra Lund-Nilsson published when she was working together with Casey and Pronk, where she did a comparison of human and mouse stem and progenitor cell composition, and there are evident sort of similarities. So for instance, early lymphoid cells, precursors for those go down in humans. They go down also in mice. The increase in stem cell numbers that Hartmut was alluding to, you can see that in humans. You can also see that in mice. But there are differences also. So, whereas human bone marrow is characterized by, elevated numbers of these megakaryocytic erythroid precursor cells. So in the mouse, we cannot really see that for the erythroid precursor cells, although we can see, increase in megakaryocytic precursor cells. So the composite view then, so just looking at sort of similarities here, so I think that we can conclude that we have an expanded stem cell compartment. There is some type of bias, a erythroid megakaryocytic bias, and the idea here has been that this reduced lymphopoiesis in favor of myelopoiesis somehow could be linked to myeloid malignancies. Now, how has these sort of concepts, how have they actually been sort of established? Now, looking at how you can establish intrinsic changes at the level of stem cells. So I think that for many years this was an experiment I liked quite a lot. This was done by Hartmut Geiger in my lab. So he isolated stem cells from either aged or young mice, and then he was using the congenic CD45-1 and 2 systems. And then he was evaluating these type of cells in an F1 host, which expresses both of these markers. So, the beauty of the experiment, I think, is that it allows you to assess, stem cell contribution from either aged or young HSCs in the same host. In this case, it was young recipients. Now, I should say one point here is that because of the elevations in stem cell numbers, so this experiment here was actually corrected for in terms of cell numbers, so that we transplanted 4 times more of the aged HSCs. Now, despite this, so long-term after transplantation, looking at the contribution to the individual lineages is, quite pronounced decreases observed in the B and the T cell lineages, so adaptive immunity. Whereas, by contrast, the myeloid contribution is much higher. So I think that, with these type of experiments, so with them we can conclude that we have an alteration in lymphopoiesis with a reduced lymphopoiesis. We can also establish that we have a reduced repopulation capacity because we can merely look at sort of the overall levels that our HSCs contribute to. But this idea that we have sort of a myeloid bias or a myeloid overrepresentation, so that I think maybe is something that is worth to discuss, whether we have that or whether actually the myeloid readout that we observe is actually mainly a consequence of reduced B and T cell contribution, because as I told you, in this case here we actually transplanted 4 times the amount of aged HSCs, but we can only translate that into about a 2-fold difference in the contribution to the myeloid lineage. So meaning that actually the HSCs from the aged mice contribute less also to these lineages. Now, then coming into the question of reverse of aging, whether that is possible. So basically, we can structure it like this: that if we have a stem cell that is able to differentiate in a balanced fashion into a couple of different lineages, but also maintain itself, we have the aging situation where we have maybe a bit more contribution to the stem cell pool, more to an individual lineage, but less of another one. And so, of course, it's this situation here that we would actually like then to reinstate and sort of correct to some extent back to this. So Martin Wallerstedt in my lab, so he took an approach many years ago now, where he used an IPS reprogramming approach where he started the experiments by isolating aged stem and progenitor cells. He retrovirally infected these with reprogramming factors, the standard ones from Yamanaka factors, allowed IPS formation, and then harvested these IPS cells, implanted them into new blastocysts, and then assessed whether or not it was possible to obtain any chimeric animals. And having taken advantage also here of the chondrionic CD45-1, CD45-2 systems, it was also possible to directly assess the contribution of these aged IPS cells to hematopoiesis. And so here the experimental paradigm allows you to study not only the primary chimeras, but also you can extract bone marrow from these ones, transplant it into new mice, and study the stem cells capacity following regeneration. And this is just some data from that work, so focusing on yes, the lineage output. And so actually in of the sort of two well established clones in this setting, so we found actually no evidence that they would deviate from that of the endogenous blastocyst or young cells. So meaning really that despite that the starting or parental cells in this case here was substantial age, 2 years of age at the time of IPS generation, propagation, and then implantation, and then study half a year or so after the generation of mice. Focusing in on the T cell lineage, and I think that Hartmut introduced this quite nicely also in his presentation, that actually the reduced T cell output in connection to age is not only an effect of the involuting thymos. I think that this experiment here shows that quite well. So in these experiments we were evaluating the production of naive T cells as evaluated by T cell receptor excision circle PCRs. So when you transplant HSCs from either young or adult or aged sources, as you can see that the output of TRK positive T cells in the normal age setting is extremely low, similar as you actually see just in the steady state native situation. But following evaluation of these reprogrammed cells, you can see that actually the numbers of TRKs produced was more or less back to the same levels as that of young or blastocyst derived. Similarly, if you now assess the sort of stem cell quality in terms of quantitative output, so the red and the green lines here would just indicate the repopulation activity on a per cell basis of a young or an aged HSC. And so you can see that actually in this case here, these two IPS lines, they were actually sort of comparative to the young cells in regenerative output. Now, this was all fine. So in a way, it sort of provided us with some type of evidence that it is possible to take chronologically aged cells, reprogram them, and then you can generate a blood system with more young properties. But of course, there are some alternative interpretation of this, and so in the simplest form, so I think that we can imagine that either we have a pool of young HSCs that in the aging scenario has caused an alteration. When we do our reprogramming, we can actually convert this fully back. But there is of course also the possibility that when we do these type of experiments, that there is a fraction of the cells that are actually unaffected by age, and that the reprogramming approach would select for those type of cells, meaning that actually those cells never displayed any functional signs of aging. And we were thinking quite a lot for a long time on how one could potentially approach this, and eventually with the advent of cellular barcoding, we thought that we actually could come up with a way to approach it more directly. So basically the way that this works is that we have a number of stem cells that are individually marked with a unique DNA tag. And then we can transplant many of these marked cells into an individual mouse. We can harvest different type of cells. And then we can assess the barcode contribution, sometime after transplantation and see, so do we see contribution of all these originally marked cells to all these different lineages. And we could then, of course, imagine that if we have a sort of a balanced output, we would get contribution from all our barcoded cells. But if we would have a sort of a lineage skewed scenario, we would see these distortions in the output, so that certain barcodes would dominate and certain barcodes would be absent from certain lineages. Now, in essence, this is the same type of experiment as you do when you do a single cell transplantation, but the key advantage here is that it allows you to evaluate a lot of the clones at the same time. Now, by combining then the barcoding approach with this approach that I described to you previously on the blastocyst IPS generation and blastocyst generation, so we also took advantage here of some advances in reprogramming, where at this time transgenic mice had been available where you could induce the reprogramming factors by simple addition of doxycycline, so which would greatly enhance the IPS generation frequency. So we then took the approach to age these reprogramming mice, go through the whole experimental procedure again, and then assess at the clonal level now, what do we actually see. Now, this is a very busy slide, but in essence, what the point of this one here is that really, I think that these experiments changed at least my view on some of the aspects of HSC aging and the contribution. So if we look at the lineage competence, that is the, what type of mature cells aged HSCs can contribute to, you can see that it's actually not a big difference compared to young, with one exception, and that was for the T cell lineage, where actually we could observe that there were way fewer aged HSCs that were able to contribute to the T cell lineage. But the striking thing was really the magnitude of contribution, and that applied more or less to all lineages except for the T cell lineage, because in the cases where we could observe a robust T cell contribution, so from these few clones here, the clone sizes were actually, these were actually very robust stem cells. Now, I think that we were not the first and to show that actually this happens. So this is a couple of studies, but in essence, so what the experiments showed was that the number of truly multipotent stem cells in the aging scenario, which is actually represented by this here, this is a type of cell that is severely underrepresented with age. Now, having all the sort of clonal data, that of course also allowed us to extract the barcodes. So going into the bone marrows of these analyzed animals, generating IPS cells, and then sequence from each generated IPS clone, sequence the barcodes, and then being able to retrospectively go back and see what actually did that clone contribute to. Now, looking again in the T cell lineage, so again with the 5 assessed clones in this situation here, so even though the parental cells from all of these 5 clones didn't produce any T cells in the primary recipients, so upon the IPS reprogramming and even following secondary transplants of those primary chimeras, so we could see that actually we now had a T cell generation that was more or less indistinguishable from that of young mice or from young HSCs. Looking at the regenerative properties of these clonally derived IPS cells, so again, so we can really not see any difference in the sort of frequency of HSCs, so this pronounced increase that we normally see with age, but so this is back to normal even after transplantation. And looking at gene expression, so here looking at single cell gene expression, looking at a panel of known age-regulated genes and just the PCA analysis of these genes. So we can see that actually the iPS-derived HSCs and the young HSCs, they cluster very closely together and quite distinct from that of aged or mid-aged HSCs. So I think I'll sum up here, and sort of the key points. What I wanted to make here was really that we should distinguish between functionally and merely chronologically aged cells. In fact, our results revealed the presence of what would say aged HSCs, although they are more rare, but also in young mice. But from a more conceptual point of view, it seems as though at least some aspects of rejuvenation of function, functional rejuvenation is possible, which would then in essence also mean that DNA damage does not seem to account for all aspects of HSC aging. Okay, and the work that I've been presenting today is work mainly by a very talented PhD postdoc, Martin Wallerstedt, in my lab, and of course, all the great collaborators that we've had. Thank you.
Thank you so much, David, for a great talk and really inspiring. So now we will begin with the question and answer portion of this presentation. And I do have already a couple of questions, so I'm going to start that. First one is for Hartmut. So about the casein rejuvenation effect. So it's really interesting how this is also affecting the immune system for vaccination response. Can you speculate, or do you have any data about the effect of casein for leukemic stem cells or initiating leukemic stem cells?
Yes and no. So okay. Can you hear me? Yes.
Yes.
Collaborators in Cincinnati have studied how casein will act on leukemic stem cells, and I guess it comes down to type of leukemic stem cells and how they are organized. To best establish their differences, but some of them start to become disorganized, and there is a response of casein also for leukemic stem cells. So that's actually a very promising approach. You could also argue that probably leukemia pre-ages some of these stem cells. But again, it's not our work, it's from collaborators in Cincinnati. Jim Malloy and Yi Cheng have worked on that.
And I also have another question still related to this first question. Did they have a look at the polarity of these leukemic stem cells? Like
they had a look, and we had initially a look again on some types of leukemic stem cells are apolar, but it's probably not all type of leukemic stem cells. It might also come down to the type of oncogene they are driven by.
All right. So we do have one question for Hartmut from the audience. So the question is, what is the effect of casein on CDC42 GTP activity in young mice?
So casein inhibits CDC42 activity no matter how old you are, and if you give that to young mice, you also will inhibit the activity to very low levels in young mice or young stem cells primarily. So we don't have the interesting part though is because let's say if you do it short term and transient, they don't mind too much. So which means if you then ask the question, we have published on that again, do you look similar to a still young stem cell? Yes, you do, if you just have a short-term treatment. And also so far toxicology studies on casein given to young mice would indicate that at least for the pharmacodynamics and kinetics they have in vivo, it's pretty short-term, it does not seem to affect function in a young mice. So transiently, when you inhibit CDC42 activity in young stem cells or young mice, it does not seem to affect their proper function.
Thanks. And I do have a question also for David. Regarding the reprogramming of HSCs in your system. So you have done extensively epigenetic studies showing that the reprogrammed ones looks similar to the young hematopoietic stem cell. So have you deep into this mechanism in the sense, did you look at any epigenetic marks that are recurrently changing and driving this reprogramming in your clones? Did something popped up out of your epigenetic studies?
No. So I must say that we have not. So the unfortunate thing with the approaches that we have taken is that the numbers of cells that you actually can obtain for study is extremely rare. We as I showed you in the end, so we took single-cell gene expression as a proxy for the changes that occur. But really, I mean, we end up here with a few hundred cells, maybe at best, from each individual animal. So I think that although with the advances in technology developments, so probably it will be possible in future on to try to begin to be a bit more sort of mechanistically oriented, but no, really not. So one thing I can say at least with the approaches we took on the when we made the iPS cells using the retroviral approaches. So we were looking into the telomere length, and actually one thing that you can see there is that the reprogramming associates with a quite heavy telomere elongation. Mm-hmm. Which is stable. And you need really genetic dilution in order to shorten it. So we are not the first to have reported that, and we did not really assess that in the second approach. And I maybe think that it doesn't have so much to do with it, given the long telomere length of mice. But that is really one thing that we know happens with the reprogramming, is that we get an elongation of telomeres, probably because of reactivation of telomeres.
All right. Thank you.
I got a question for, do you have more questions? No, I got a question for David, actually. Can I ask a question?
Of course, go ahead.
Hello?
Of course.
Yeah.
Okay,
sure.
Good. So, David, what do you think is the purpose of having, or is there a purpose of having already, let's say, functionally aged stem cells in a young mouse, for example? The data is based on mouse, mm-hmm. Because your data would support that, and our data, because we have a level of cells that are already, let's say, apolar when they are young, and we would assume that these are functionally more aged, mm-hmm. than the other ones. So what do you think is the reason behind it? Is there any reason behind it? Is there a purpose for that?
Well,
That's the question.
I've been thinking quite a lot on that. So whether there would actually be a purpose of having these type of aged cells, and I must say that if it was up to me, I would prefer not to have them around. But of course, they might actually be a consequence of any type of aging process in the blood system. So, for instance, if we have a bit more one of these models which is based more on successive generation from HSCs, that you can imagine that you have certain HSC clones that will be more heavily utilized at a certain age, and of course the progeny of those or their stem cell relatives would somehow display this earlier on. I think that is still something that hasn't really been established. The relationship of sort of proliferative aging and the aging phenotypes we see at the HSC level. So I know that there are, you can of course evoke many of the aging phenotypes by serial transplantation or by some other type of stressors. And in that case, there is of course a clear link between stem cell proliferation and the manifestations of an aging function. In a normal situation, of course, it's quite well established these days that stem cells are not super actively contributing, although in our hands, we can always see contribution of HSCs in a sort of native hematopoiesis. it's not at all to the levels that you see following a transplant or cytostatics. So, maybe it is actually more of a sort of a side effect, so to say.
Okay.
So actually, I have a question for Hartmut, if that is okay.
Yeah. Nice. I like that. Go ahead.
So in your work, in the end of your presentation, so when you study sort of immune cell aspects, so you were using RAG1 deficient mice as hosts.
Yes.
And not sort of wild type hosts or other type of immune deficient hosts. So the reason I ask is that we just completed one study where we were comparing different type of immunodeficient hosts, completely different product, leukemia-associated product. But what we observed in terms of lymphoid reconstitution was actually that RAG-deficient hosts were very intolerant for B-cell reconstitution. So compared to RAG2 gamma chromatin deficient hosts, for instance, where you get really robust levels of B cells. So is there any sort of, can you sort of imagine that there is a difference here? Because of course also the RAG deficient host has never had an immune system, and there are not even traces in a mouse like that of an immune system. Whereas if you would do it in a wild type situation, even if the mice are conditioned, of course it's very difficult to target, at least some of, memory type T cells. And do you have any insights into that? Does it own, is it?
So first of all, we took the RAG mice because you can clearly show radiation. Even if you rate animals very strongly, you have still a strong contribution of recipient T cells that are radiation resistant. So you will always end up in a mixture between, let's say, usually the memory type of T cells in this background, even if you transplant fresh cells into. So at the end, you will not clearly know which one is coming, the response is coming from which cell. So this was the major concern we had. So in RAG mice, you don't have that because you don't have T and B cells. As you can see, they nicely constitute T cells, and we only saw, we saw a strong B cell contribution too, which we did not compare to other immunodeficient animals. So it might be less or more, I definitely don't know that, but it's there. And we're still in the process of looking at the functional contribution in the system of B cells to immunization. So the one we tested currently is primarily a T cell based one, because it's a little bit easier to measure in the system, and it responds nicely to DNA vaccination, which is a very straightforward measurement of immune competence, at least of the T cell branch. That's all I can say so far. We have not checked the B cell part in that great detail compared to the T cell part, but it's due to people in immunization world for these DNA vaccines like T cells a lot. And it seems to work very nicely in establishing a T cell response if you look at the rejuvenated stem cells, which I was very surprised the robustness and the level you can get in the system. I agree. Good.
All right. So it seems like we don't have any more questions. So I'd like to close the session webinar for today. And thank you all for participating in today's webinar, and especially a special thanks for our presenters, David and Hartmut, for today's session, for the great talks. And just a reminder to all the attendees, please complete the evaluation for today's event that you will receive by email, because your feedback and input are important and appreciated as further develop ISEH webinar programming. And thank you so much again for attending, and have a great day.
Okay, you too, all participants. Have a great day. Bye.
Thanks all.